In order for the nervous system to do its job of receiving, processing, and sending information, it needs a way to communicate with itself. It needs a way for one neuron to send information to another neuron. And that's going to occur at a synapse. So a synapse is actually a connection between a neuron and its effector cell. So this can go from neuron to neuron, or from neuron to a muscle cell, or from a neuron to a gland. We call the neuron that is sending the signal the presynaptic neuron. So it's the neuron where the signal travels through before reaching the synapse. And then the neuron that gets affected, we call the postsynaptic neuron. It's the neuron after the synapse. So again, the synapse is the connection between the two neurons. We have our presynaptic and then our postsynaptic. So the presynaptic neuron is the neuron sending the signal. The postsynaptic neuron or cell is going to be the one receiving the signal. In the peripheral nervous system, this could be another neuron, or it could be a muscle cell, or it could be a gland. Most neurons are going to be both presynaptic and postsynaptic. They receive information from other neurons and send information to other neurons. Our types of synaptic connections, we typically are going to have either axodendritic or axosomatic. So axodendritic is when we have the axon attaching to the dendrite. Remember, the dendrites are going to be these receptive processes on the neuron. And then axiosomatic is when the axon is going to attach to the soma or cell body. The cell body is also going to be a receptive part of the neuron. Much less common is when you have the axon to axon synapses, what are called axo-axonal. So usually we are going to have synapses that go attached to dendrites or directly to the body of the neuron. Remember, the signal is going to be summated inside of the body of the neuron. Synapses come in two main types, what are called chemical synapses and electrical synapses. Chemical synapses are much more common. This is the classic synapse. This is where a neurotransmitter gets released across a synaptic cleft. There's not a direct electrical connection. The electrical signal travels down the axon, gets converted into a chemical signal, and then that chemical signal gets converted back into an electrical signal in the postsynaptic cell. In an electrical synapse, there's a direct connection through gap junctions, but we pretty much only see that during neurodevelopment, when the nervous system is being formed. So our chemical synapses are our most common types of synapse. This is our classical synapse, and it's made up of two parts. We have the axon terminal, that's the end of the axon of the presynaptic neuron, and then we have the receptor region on the postsynaptic neuron, usually on a dendrite or on the cell body, the soma. At our axon terminal, the axon terminal, also called the terminal bouton, is going to be filled with synaptic vesicles that have neurotransmitter. So separating the two, we are going to have this synaptic cleft filled with fluid. Note this synaptic cleft is actually really, really tiny. There are proteins that actually span this cleft to hold the axon terminal very, very close to the postsynaptic neuron. And then on the postsynaptic neuron, we are going to have receptors for the neurotransmitter. So we have the presynaptic neuron with the axon terminal, which is going to have synaptic vesicles filled with neurotransmitter that get released into the synaptic cleft, which are then going to bind to neurotransmitter receptors on the postsynaptic neuron. So in order to send a signal at a chemical synapse, we're going to have an action potential traveling down the axon of the presynaptic neuron until it reaches the presynaptic neuron's axon terminal. At the axon terminal, it's going to cause the opening of voltage-gated calcium channels. And calcium is going to flow into the axon terminal. The presence of calcium is going to trigger the synaptic vesicles to fuse with the membrane of the axon terminal and release the neurotransmitter into the synaptic cleft. The neurotransmitter is going to diffuse across the synaptic cleft. This happens very quickly because this is actually a very small space, and bind to the neurotransmitter receptors on the postsynaptic neuron. That is going to typically cause these neurotransmitters to open. These are often going to be chemically gated ion channels. So when they open, they are then going to cause a graded potential, one of those postsynaptic potentials, PSPs. And that can be either an EPSP, an excitatory postsynaptic potential, making the cell more likely to fire an action potential, or an IPSP, an inhibitory postsynaptic potential, making the cell less likely to fire an action potential. Then we have to stop the signal. And that can happen in one of three ways. We can have reuptake, where either the axon or nearby astrocytes have these pumps that suck the neurotransmitter back up, or we can have enzymes that break down the neurotransmitter. We saw that at the neuromuscular junction, which is a type of synapse, where we have the acetylcholine esterase break down the acetylcholine, and then also we just have diffusion. Eventually, the neurotransmitter just drifts away and becomes too low of a concentration in order to continue to send the signal. So, action potential travels down the presynaptic neuron axon, reaches the axon terminal. It causes the opening of voltage-gated calcium channels. Calcium flows into the axon terminal, causes synaptic vesicles to fuse with the cell membrane of the axon terminal, releasing neurotransmitter into the synaptic cleft. Neurotransmitter binds to neurotransmitter receptors, which are often chemical gated ion channels. The chemical gated ion channels open. This causes a graded potential, either an EPSP or an IPSP, and then we need to shut down the signal either by sucking the neurotransmitter back up with reuptake pumps, breaking down the neurotransmitter with enzymes, or just letting it diffuse away. At an electrical synapse, again, these are not very common in adult brains. The neurons are directly electrically joined together by gap junctions. So the communication in this case is very quick because we don't have to translate from electrical signal to chemical signal, then back into electrical signal. Instead, we just have the direct flow of charged particles, allowing that electrical signal to spread very quickly. This also allows for bidirectional communication, and we do use this in some brain regions. So we see this a little bit in the hippocampus. We see this in some brain regions that are involved in eye movement, but we primarily see it in embryonic nervous tissue.